Welcome to the Sand Science Experience podcast, where we explore the latest and most exciting papers and preprints in science. Before we jump into today's discussion, please remember to hit that like and subscribe button and share this deep dive with your friends and family. It really supports the Sand Science Experience and allows us to keep analyzing this cutting-edge research. Today, we are undertaking a deep dive into, well, something quite astonishing in scientific automation, an AI agent called Cosmos. This system isn't just about streamlining tasks. It's designed to conduct what amounts to months of human-level research in just a single autonomous run.
Yeah, what we're looking at here is potentially a fundamental shift. Cosmos is probably best described as an end-to-end AI scientist. You basically give it a massive public data set and a really open-ended objective like, say, find a novel mechanism for Alzheimer's progression, and it just starts working. It runs these iterative cycles of literature search, hypothesis generation, data cleaning, analysis, and importantly, it does this successfully across wildly different fields. We're talking neurobiology, materials science, metabolomics.
Okay, so our mission today then is to explore exactly how Cosmos achieves this kind of scale and depth, what its actual verifiable outputs look like, and maybe touch on what the implications are for, well, for us, the humans who currently do this kind of research.
Right. And I think to really appreciate the innovation, you have to look back a bit. Previous attempts at these autonomous scientific AI agents, they sort of hit a wall because they quickly lost what you might call coherence.
Yeah.
Scientific discovery is this long, iterative conversation. You read a paper, you form a tiny hypothesis, you test it against some data, and then you use that result to guide your next search. Traditional AI systems often couldn't really maintain that long thread of logic. They failed pretty quickly.
Okay, let's unpack this then. So the core breakthrough that allows Cosmos to sustain that thread is something they call its structured world model. Is that right? So should we think of this model as like a constantly updated central blueprint, one that holds the research objective and all the discovered facts so far?
Exactly. It's this centralized coherence that allows it to share and synthesize information really efficiently between its specialized workers. Specifically, there's a data analysis agent and a literature search agent working together.
Ah, I see.
And that persistent coherence, that's what translates into this massive scale. Cosmos can execute an objective for up to 12 continuous hours. That means pushing through over 200 agent rollouts, and each one is potentially a step in the scientific method per cycle.
12 hours straight, and when you look at the sheer volume of work being processed in that time, that's where the comparison to human labor really starts to sink in, doesn't it?
Oh, absolutely. The output is, well, it's truly staggering. In an average run focused on a specific objective, Cosmos executes approximately 42,000 lines of analytical code. A lot of that is self-generated, by the way.
Yeah.
And it reads, synthesizes, and incorporates findings from about 1,500 full-length scientific papers.
1,500 papers.
1,500. Yeah. And the human collaborators, the scientists working with it, they estimated that just one single 20-cycle run performed the functional equivalent of 6 months of dedicated research time for an average human scientist in their lab.
6 months. 6 months of digging through literature, scripting data analysis, iterating on hypotheses, all compressed into 12 hours. I mean, that just changes the job description overnight, potentially. Imagine being able to offload the, frankly, drudgery of reading and synthesizing those 1,500 papers to a machine. That frees up the human scientist to focus purely on the interpretation, the validation, the bigger picture thinking.
It creates a completely new level of acceleration. And what the sources found, crucially, is that the number of valuable, actionable scientific findings generated by Cosmos actually scaled linearly with the number of cycles they tested. So more work equals proportionally more discovery. It's not hitting a wall.
That efficiency is incredibly compelling, but, the absolute fundamental requirement in science is rigor and traceability. If an AI claims something, how do we confirm it didn't just hallucinate or follow some spurious lead? That's critical.
Absolutely critical. And
Yeah.
that requirement was actually built into the system's architecture from the ground up. The traceability is absolute. Every single statement, whether it's an assertion about data correlation or maybe a historical claim about a biological pathway, is cited. The citation points either to the exact line of code within the run's Jupyter notebook so you can see the calculation, or directly to the primary literature source it used to make that assertion. The internal process is basically a transparent, auditable record.
Okay, so we know where the claims came from. That's good. But how accurate are they? Did the authors actually validate these findings externally?
They did, yes. They had independent scientists evaluate a selection of, I think it was 102 representative statements pulled from across the reports. The overall accuracy rate was pretty strong.
Yeah.
79.4% of all statements were classified as correct.
Wow, nearly 80%. That's a highly impressive reliability score for an autonomous system trying to do novel discovery. But, okay, this raises an important question then. If the system is reliable, say, 4 times out of 5, where does it tend to fail? What's happening in that other 20%?
Yeah, and this is where the distinction between a powerful calculator and well, a scientist truly matters. The accuracy varies significantly based on the type of statement. So statements based purely on data analysis, meaning, high fidelity computation, running statistics, generating complex code, those were 85.5% accurate, very reliable. But where the accuracy really dipped, quite significantly actually, was with statements that required interpretation and synthesis, connecting the data dots with external literature. Those statements were only 57.9% accurate.
Ah, okay. So just over half right on the interpretation side. Given that its interpretive accuracy dips below 60%, should we really be comfortable calling it a scientist? Or is it more like the world's most powerful, fastest data engine? I mean, the data analysis seems impeccable, but that high-level judgment, connecting disparate facts, assessing scientific merit, that still seems to absolutely require the human in the loop, right?
Exactly right. The AI excels at the exploratory statistics, generating the paths, doing the heavy lifting. But human intuition, critical judgment, maybe even scientific taste, those are still absolutely necessary to evaluate the meaning and the merit of those results before you commit lab time and resources to follow them up. So the system removes the drudgery, the human provides the wisdom, essentially.
Right. And we see the quantitative reliability in action in one of their examples, Discovery 1. Cosmos was given this complex mouse brain metabolomics dataset, and it managed to reproduce a really critical, yet still unpublished, finding about hypothermia-induced neuroprotection.
Yeah, and the detail here really underscores its computational power. Cosmos identified nucleotide metabolism as the dominant pathway altered by the hypothermia. Specifically, it noted the activation of this energy-efficient process called the nucleotide salvage pathway. Now, when the human researchers compared Cosmos's log-10 fold change estimates for the top 15 metabolites against their own manually calculated figures, the results were almost indistinguishable. They achieved an R-squared value of 0.998.
An R squared of 0.998 is, I mean, for all intents and purposes, that's a perfect match. That confirms that when you give it structured data, Cosmos can analyze, process, and quantify findings with just mechanical precision. It gets the numbers right.
Exactly. And if we connect this back to the bigger picture, the reason Cosmos can jump between domains so easily from brain chemistry to material science is that its underlying agents are general purpose. It's truly domain agnostic. So we can jump from the delicate chemistry of the brain right into, say, industrial material science without really skipping a beat.
Which brings us neatly to Discovery 2. This focused on the really complex fabrication environment for perovskite solar cells. Cosmos was tasked with analyzing fabrication data to figure out which environmental factors were most influencing the cell's efficiency.
Yeah, and this environment is notoriously complex. You've got temperature, solvent partial pressure, humidity, all interacting in subtle ways. Cosmos was able to isolate thermal annealing humidity as the single most dominant determinant. It essentially acts as a fatal filter. It found that device failure consistently happened above a critical threshold, about 60 grams per cubic meter of absolute humidity.
So it didn't just find the dominant variable, it actually quantified the precise threshold where things go wrong.
Precisely. And furthermore, it identified something new. A previously unreported linear decrease in something called short-circuit current density, or JSC, that correlated with rising dimethylformamide, that's DMF solvent partial pressure, during the spin coating stage. This was a subtle, specific observation that the human researchers looking at the same data had actually missed, and it was later independently validated in the lab.
Okay, so it's impressive enough that Cosmos can reproduce known complex findings and find these novel, testable relationships. But the paper also says it proved its ability to actually develop new methodology, not just use existing ones.
Yes, Discovery 6, which came from Alzheimer's disease proteomics data, is a fantastic example of this. The objective was pretty broad. Sequence the chronological events leading to tau accumulation in the brain. Now, to achieve this, Cosmos independently proposed using a novel analytical approach, segmented regression. This is a statistical method often used in fields like economics or data science, maybe not standard in basic neuroscience. It proposed using this to find a quantitative breakpoint in the decline of cellular pathways along this concept of disease progression pseudotime.
So instead of just charting a kind of steady decline, it suggested a way to pinpoint exactly when a specific decline really started to accelerate or change trajectory.
That's exactly right. And using this novel method it proposed, it determined that the decline of the extracellular matrix proteins ECM had a specific breakpoint at 0.58 units of pseudotime. This provided strong evidence to temporally place ECM failure downstream of the transition to cells becoming tau positive. That dramatically helps order the complex progression of cellular dysfunction in Alzheimer's disease.
Okay, this is where it gets really interesting, though, moving beyond just validation and methodological novelty. Discovery 7 seems to demonstrate Cosmos's ability to uncover clinically relevant biological mechanisms that were apparently not identified by the original human research team working on that very same data.
Yes, this involved digging into really complex single-nuclei RNA sequencing data. The goal was to understand why neurons in a specific brain region, the entorhinal cortex, or ENT, are uniquely vulnerable in aging and Alzheimer's disease. Cosmos discovered this coordinated mechanism. It actually labeled the prophagocytic axis activation.
Yeah.
I probably need to slow down a bit here to appreciate the biological failure it uncovered.
Okay, let's simplify that complex axis then. What's the fundamental problem that Cosmos identified? What's going wrong?
Well, the issue is essentially a two-part system failure that results in the neuron basically getting eaten alive by the brain's immune cells. So, first, vulnerable ENT neurons showed this systemic, age-related downregulation of certain proteins called P4-ATPase. Flippases, one example is LTP10A. Now, flippases are basically responsible for maintaining the neuron's membrane asymmetry. They keep the inside stuff distinct from the outside stuff at the membrane level.
Okay, so the neuron is literally forgetting how to hide its internal vulnerability signals from the outside world.
That's a great way to put it. When these flippases fail, it leads to increased exposure of a molecule called phosphatidylserine, or PS, on the outer surface of the cell membrane. And PS is universally known in biology as the eat me signal. If a cell displays PS, it's telling the immune system, I am defective, please clear me away.
Right. And what's the second part of this system failure then?
Well, simultaneously, the microglia, which are the brain's resident immune cells responsible for this kind of clearance, they showed an upregulation of the specific genes involved in the PS-sensing phagocytic pathway. So basically, the microglia learned to read that eat me signal better, just as the neurons started flashing it more often due to the flippase failure.
Wow.
The conclusion is pretty clear, quite stark. The age-related failure of these specific ENT neurons to maintain their membrane asymmetry makes them a targeted meal for microglial clearance. This provides a powerful, novel, and importantly, testable mechanism for why the entorhinal cortex is so vulnerable in Alzheimer's disease.
That is, well, that's a phenomenal piece of autonomous discovery. The system integrated these vast single nuclei datasets, identified complex gene patterns, connected those patterns to known biological functions using its literature search, and then generated a coherent, testable clinical hypothesis, all without direct human guidance on that specific mechanism. It really shows that even in these incredibly rich, highly curated data sets that experts have already pored over, there are still overlooked connections just waiting to be made.
It absolutely accelerates the entire cycle of hypothesis generation, potentially from months down to mere hours.
So wrapping this up, what does this all really mean for us? Cosmos seems to provide this ultimate acceleration loop for science. It transforms these mountains of raw data into testable, citable hypotheses in what amounts to months equivalent time, all while maintaining complete quantitative rigor. It looks like a tool that can rapidly reproduce findings, validate new methods, and, maybe most excitingly, uncover truly novel biological mechanisms hidden in plain sight within existing data.
And maybe a final thought for you, the listener, to consider. Cosmos demonstrates quite powerfully that truly novel, potentially clinically relevant discoveries like that entorhinal cortex mechanism are already sitting there, hidden, maybe dormant, in existing public datasets that human scientists have already published and perhaps moved on from. So if one AI run can genuinely perform 6 months of human work, how quickly could we potentially reanalyze the massive backlog of existing scientific data worldwide to unlock all those other hidden insights that are just waiting for discovery?
That is indeed the defining question, I think, driving what could be the next era of scientific discovery. Thank you for joining us for this deep dive into the latest and most exciting papers and preprints in science, only here on the Sand Science Experience. Don't forget to like and subscribe to keep supporting the show.